Hey guys, welcome back!
We're the Helicase Homies back at it again, but this time with DNA replication.
First, let's start off simple. As many of you know, your DNA is from half of each of your parents, meaning that genetically you are 50% of your mom and 50% of your dad. In scientific terms, this is called semi-conservative replication. Semi-conservative DNA replication emphasizes how the offspring of two parents will have an equal number of genes from each parent. Therefore, all parental genes are passed off equally to offspring. This mechanism was first discovered by Matt Messelson and Frank Stahl. These two scientists provided evidence for semi-conservative replication through their testing of nitrogen-15. Their experiment with nitrogen-15 illustrated how parental DNA was passed equally from both parents to offspring.
There are many other types of replication, but none of which us humans use to replicate our DNA. Two other types of replication are the conservative replication model and the dispersive replication model. In the conservative model of DNA replication, after the first round of replication has occurred, half of the helices are from the original DNA and the other half of the helices are new. During replication round 2, each helix is completely copied. The dispersive DNA replication model is when the DNA helix breaks apart into many fragments. These pieces will be the template for the next round of replication. This replication method results in a varying amount of old and new DNA.
Now, on to the good stuff. So how does DNA really replicate itself, making everyone genetically different from each other? There are three main steps to DNA replication. The first step is strand separation. Strand separation is the first step in DNA replication and will determine the rest of the DNA replication process. In order for DNA replication to begin, the DNA strands cannot be attached to each other and must be unwound. The strands will separate from each other at a site known as a replication origin. Within strand separation, there are two different enzymes that will take on different jobs. The first enzyme that will appear during replication is Helicase. Helicase works the most proficiently when it is found within larger groups. It will help bind your replication origin so that strand separation can take place. Helicase will help activate strand separation as it unwinds the nucleic acids found within DNA strands. Important reminder, strand separation always occurs in the 5' to 3' direction. The other enzyme that has a job during strand separation is known as topoisomerase. Topoisomerase is responsible for cutting the phosphodiester bonds that help keep strands attached. These phosphodiester bonds are found near the replication fork. Topoisomerase will help relieve tension, allowing the DNA to unwind. Along with enzymes, there are also proteins known as SSBs, single-stranded binding proteins. SSBs will only appear after strands have been separated. SSBs are responsible for prohibiting the newly separated strands from binding back together and reversing strand separation. The strands will now be separated and have a bubble in between them that is referred to as the replication bubble, where the new DNA or replicated DNA can be found.
The second step of DNA replication is known as building complementary strands. Think of it as if you were building a building that looked like another building. Building complementary strands starts with the replication fork. The replication fork opens, but the DNA polymerase is unable to bind to anything on the DNA strand. At this point, nucleotides are added to the strand. Then the RNA primase will create an RNA primer sequence so it matches RNA nucleotides to the single-stranded DNA. Then the RNA primer sequence is recognized by DNA polymerase III, which then adds nucleotides to the RNA primer, therefore building a new DNA strand in the 5' to 3' direction. There are two strands of DNA that are created. Because DNA polymerase builds in the 5' to 3' direction and towards the replication fork, one strand is able to replicate continuously. This strand is called the leading strand. The other strand is called the lagging strand, which is replicated when DNA polymerase III moves in the opposite direction from the replication fork, so it is built piece by piece. Both the leading and lagging strands are called Okazaki fragments, and they start with their RNA primers. RNA primers are removed and DNA is placed by DNA polymerase I, which replaces the primers. The final nick is closed by the DNA ligase protein. It creates a phosphodiester bond between the Okazaki fragments.
And the final, if not most important and vital step of DNA replication, dealing with errors. Dealing with errors is so vital as it can determine whether harmful, deleterious, neutral, or no mutation will arise. In order to deal with errors, DNA polymerase I and II will assist with repairing the errors that DNA polymerase III may make during DNA replication. DNA polymerase I and II will help repair base pairs that may have been damaged, or they will match nucleotides to the correct base. DNA polymerase can detect which strand has been newly replicated and which is the parent strand, so that the wrong strand is not replicated for another generation of replicated DNA.
But there are also many more complex and in-depth ways that your body ensures that all errors made during DNA replication are corrected. These processes include mismatch repair, nucleotide excision repair, base excision repair, and double-stranded break repair.
Everyone take a deep breath because we're going deep diving into error correction. First, MMR. Mismatch repair is the first DNA error correction process that may occur. This process occurs when a nucleotide is paired with the incorrect base pair. To ensure that the mismatch does not become permanent, mismatch repair occurs. The MUT-S protein is able to recognize a mismatch. Then ATP-bound MSHs recruit the MLHs, which link the distant transition to the mismatch recognition signal. The removal of DNA that contains the incorrect nucleotide sequence occurs next. Finally, the remaining DNA strand acts as a template for the replicative DNA ligase to resynthesize the excision gap.
NER, or nucleotide excision repair, is a process used by many mammals to remove lesions in the DNA. Mutagens found in the environment, exposure to UV light, and several other causes create these errors. Essentially, using complex proteins, the nucleotide excision repair process detects and removes the incorrect lesion. DNA polymerase delta, and polymerase K will then remove the damaged DNA by adding the correct bases. Then finally, DNA ligase will reconnect the DNA.
The next repair process that might occur is BER, or base excision repair. The first step in this process is that damage of the bases is detected by a type of DNA glycolase. There are many different types, which each is responsible for detecting a different type of damage. Next, an AP site is created. The AP site occurs when the incorrect bases have been removed. A protein then makes a space in the DNA to ensure it is ready for repair. Beta DNA polymerase adds the correct base to fill in the gap using the correct strand as a template. Finally, DNA ligase closes the nick.
And finally, the last repair process is DSBR. DNA forms a complex by binding up to a protein called Ku, which stores proteins used for repair, such as polymerase, ligase, and other proteins. Nuclease will then remove a small amount of the ends of the strand to prepare them to join together. Then, polymerases may add nucleotides to the ends of the strands. Finally, DNA ligase is used to close off the ends. This process may occur in a different order, so the process is often referred to as flexible as the errors may vary.
Now we know that was a lot of information to take in at once.
But trust us, once you let it sink in, you will never forget it.
Thank you so much for watching, and catch us next time for protein synthesis. Bye!